Hello, my name is Lauren Hinckley, and I'm a junior pre-dental student majoring in neuroscience. In microbiology 407, I was fascinated by how the oral microbiome can impact various body systems. Later, I learned that oral microbes can impact brain health. I decided to do an independent study under Dr. Strand for Neuroscience 480 to further explore the interconnections between oral microbes that contribute to oral diseases and subsequently Alzheimer's disease. Oral health is more than just brushing, flossing, using mouthwash, and visiting the dentist regularly. Oral health and subsequently disease can impact our body systems, which are interconnected to one another, such as the heart, lungs, and gut. My research is focused on the interconnections between oral disease and how it can contribute to neurodegenerative diseases, specifically Alzheimer's disease. In order to understand how diseases can contribute to Alzheimer's disease, we need to understand some basic terminology. Oral microbiology is the study of the complex environment where microbes live, which includes various communities that microbes inhabit. Oral microbiology also includes how the microbes interact with other microbes in the communities and subsequently the host. The host is an organism that a microbe lives in or on. The oral microbiome also includes the microbial population within the mouth and looks at the genome of all the microbes to understand gene expression. This is important to understand how microbes and their communities contribute positively or negatively to health. Specialized communities that these microbes can live in are biofilms. A biofilm can grow on an abiotic or abiotic surface, such as a tooth. The microbes can attach to glycoproteins that form an acquired pellicle, which is a protein-based layer on the surface of teeth, which can provide a niche for bacteria to bind. Glucose is found in the pellicles, which can be from glucans secreted by bacteria, glycoproteins, and from the diet. This can serve as a nutrient reservoir for bacteria. Attachment to the pellicle can happen reversibly or irreversibly. Microbes can also have factors to enhance attachment. Cohesion is a type of attachment of bacteria where bacteria physically attach to one another. Then, the microbes from cohesion can then secrete EPS, or extracellular polymeric substance, which can be mediated by cohesion, and it is secreted to allow for scaffolding between bacteria and provides a barrier between the bacteria and the immune system. Bacteria can further attach in a biofilm by adhesion, which is the attachment of bacteria to the EPS. After this, a biofilm can then undergo translocation, which is the breaking off of a biofilm and adhering to other parts of the tooth, mouth, or body. Now that we know how biofilms start, let's talk about what makes a healthy oral microbiome. A healthy oral microbiome is diverse in bacterial species. The oral microbiome is the second most diverse microbiome in the human body. It can have over 700 various species of bacteria, which include firmicutes, bacteroidetes, protobacteria, and actomyces. Good oral hygiene practices can help protect your teeth and ensure that all of the food and beverage you have consumed throughout the day is off your teeth. Then, a diverse diet can also contribute to healthy states, which includes an abundance of antioxidants, flavonoids, and polyphenols that all contribute to an overall healthy state. Now that we know what constitutes a healthy oral microbiome and what contributes to a healthy oral microbiome, let's shift to how disease states start. Microbial dysbiosis is the shifting of microbial equilibrium that can have adverse health outcomes, such as promoting a disease state. This can occur from opportunistic pathogens, which are pathogens that take advantage of a condition that is out of the normal. Microbial dysbiosis can decrease the number of beneficial bacteria and their products that are made. Then there are various factors that also contribute to oral diseases. such as diets high in refined sugar that can actually increase cervical gingival fluid, which promotes more carbohydrate deposition on the tooth pellicle for bacteria to feed off of. This has a strong positive correlation to pathogenic microbial attachment. Oral dysbiosis can contribute and be attributed to obesity. As the gut and the mouth are connected, the proportion of select phyla of bacteria change in the mouth and the gut to promote more chronic inflammation. Genetics also plays a role in oral dysbiosis by contributing to having weak enamel, low immune defenses, and the production of cervical gingival fluid. Pregnancy is also attributed to oral dysbiosis through an increased amount of estrogen secretion. Poor oral hygiene can also perpetuate bacterial growth that contributes to oral diseases. There are also various microbes that are significant in dysbiosis. One of the first microbes that we see are cocci, which are circular bacteria. Another change that we see is a shift from aerobic to facultative anaerobic bacteria over time and position. Another step that we see is that facultative anaerobes shift to anaerobic populations. One of the hallmarks of a disease state, especially in oral diseases, is the presence of P. gingivalis. Promorphorus gingivalis is an anaerobic bacterium, which means that this bacteria does not use oxygen during metabolism. P. gingivalis is also a gram-negative bacterium. Gram-negative bacteria have LPS, or lipopolysaccharide, which is formed of an O antigen core oligosaccharide and lipid A in the outer membrane of the cell wall. P. gingivalis also has multiple virulence factors. A virulence factor is a characteristic or a product produced by microbes to ensure survival. Some virulence factors of P. gingivalis includes proteases, which cleaves proteins, attachment factors, which are used to attach to other microbes, or EPS, as well as lipopolysaccharide, which is on the outside of the bacterial membrane. LPS is significant in upregulating inflammation. Inflammation is the movement of white blood cells and release of chemical toxins. LPS specifically triggers macrophages to secrete pro-inflammatory cytokines. Pro-inflammatory cytokines are further upregulated by LPS. Then, part of the biofilm can actually translocate and readily adhere to other parts of the body, such as the throat, lungs, heart, and brain, to cause systemic inflammation. Other causes of inflammatory diseases, such as diabetes, obesity, atherosclerosis, juvenile and rheumatoid arthritis, incident ischemic stroke, and glaucoma, which are all precursors to Alzheimer's disease.
Pro-inflammatory cytokines in abundance produced by LPS have detrimental effects in helping to lead to cytokine-induced apoptosis, which is a signaling mechanism to trigger cell death. P. gingivalis secretes gingipains that increase the production of pro-inflammatory cytokines. Gingipains are proteases that directly damage endothelial cells that protect the blood-brain barrier. There are two gingipains that are produced by P. gingivalis, which include lysine and arginine gingipains. The blood-brain barrier contains the network of blood vessels that supply the brain with oxygen, as seen in the photo. However, the blood vessels also have endothelial cells that surround the blood vessels to protect the brain from any waste or toxic products found in the blood. This is different than the meninges, which are the coverings of the brain. P. gingivalis can travel to the brain in many ways. One is to already infect monocytes as well as damage and infect endothelium. Once P. gingivalis is in the brain, lipopolysaccharide of the bacteria can bind and attach to microglia. Microglia are the scavenger cells of the central nervous system, which activate to become macrophages that secrete cytokines and can specifically secrete pro-inflammatory cytokines. The signaling pathway that controls pro-inflammatory cytokines is highly debated. After reviewing over 50 papers and reviewing 14 papers that focused on the TLR, TREM1, and TREM2 pathways, I created my own signaling pathway, which utilized molecules that were found in most of the papers. However, most diagrams from recent studies were not done in great detail. As the central nervous system is being attacked by the microbe, let's take a closer look as to what happens in the signaling pathway from the compiled papers. TLR4 is a toll-like receptor that is responsible for initiating pro-inflammatory cytokines when the binding of PLS from a gram-negative bacterium occurs. TREM1 is also a receptor responsible for upregulating pro-inflammatory cytokines when bound to LPS. In this figure that I made on BioRender, you can see that TLR4 and TREM1 are interconnected and work synergistically to upregulate the secretion of pro-inflammatory cytokines. However, TREM2 performs the opposite function of TREM1. TREM2 is responsible for the production of anti-inflammatory cytokines, but here all we see are inhibitory pathways. LPS from a gram-negative bacterium can actually increase the transcription and translation of the TREM1 receptor in microglia in order to amplify inflammation, and it can also reduce the expression of TREM2 in microglia within the CNS. However, neuroinflammation alone is not the only contributor to Alzheimer's disease. The generation of amyloid beta plaques are also major contributors to Alzheimer's disease pathogenesis. In the Golgi body of the neuron, protein kinase A can activate one of two enzymes. Looking at the typical pathway, pKa can phosphorylate alpha secretase, which helps to cleave amyloid precursor protein in conjunction with gamma secretase, which is cleaved by the gamma secretase activating protein, splicing APP into two pieces for degradation. In the neurotoxic pathway, pKa can phosphorylate beta secretase, which cleaves APP in conjunction with gamma secretase. However, beta secretase cleaves APP incessantly. P. gingivalis helps beta secretase by secreting arginine gingipain B, which cleaves meprin beta, further cleaving APP into smaller parts called amyloid beta plaques, which is toxic to neurons. Amyloid beta plaques are toxic to neurons because these formations cause pore forming in the neurons, which results into the leakage of ions, disruption of cellular calcium balance, and the loss of membrane potential. It can also promote apoptosis, causes synaptic loss, and disrupt the cytoskeleton. Amyloid beta further causes inflammation of microglia through the TLR4 and TREM1 pathway. The abundance of pro-inflammatory cytokines also contributes to the autophagy of neurons. Furthermore, research has shown that P. gingivalis can contribute to the increasing tau phosphorylation. Tau proteins are microtubule associated proteins that are responsible for axonal transport, neurite outgrowth, and neuroplasticity. Hyperphosphorylation of tau causes neurofibrillary tangles, which inhibits the transneuronal communication, which is another hallmark of Alzheimer's disease. However, the exact mechanism in which P. gingivalis contributes to the hyperphosphorylation of tau is unknown. As we see, P. gingivalis is an important microbe that contributes to neuroinflammation and neurotoxicity. But a few microbes does not definitively cause someone to have Alzheimer's disease. The amount of P. gingivalis within the body is incredibly important. The more P. gingivalis present, the more damage is caused from virulence factors and inflammation. Our immune system can make antibodies to fight against P. gingivalis, and the amount of antibodies in the blood is being looked at as a pre-screening tool for Alzheimer's disease development. Furthermore, P. gingivalis antibodies have a strong positive correlation to total tooth loss. P. gingivalis cannot be treated with broad-spectrum antibiotics as it is very resistant. And there are other microbes within the mouth and gut that contribute to health, so we can't kill those microbes. Current treatments involve treating various symptoms of Alzheimer's disease. However, gingipain inhibitors are being looked at in human clinical trials in phase 2, actually. Lysine gingipain inhibitors and arginine gingipain inhibitors can be used as a narrow spectrum targeted treatment, unlike broad spectrum treatment, which contributes to antibiotic resistance from P. gingivalis and subsequently other bacteria within the microbiome. Overall, we looked at the habits that contribute to a healthy oral microbiome, what causes oral dysbiosis, disease progression, P. gingivalis, and how P. gingivalis contributes to Alzheimer's disease. The oral microbiome is incredibly important to maintain overall health and most importantly, contributing to brain health. Thank you so much for listening. Here are the sources that I utilized to come up with my signaling pathway for the TLR4, TREM1, and TREM2 pathway.